methods by using living organisms in order to produce different products which are useful to human beings. Okay. Now let us see the principles of biotechnology. There are two principles which biotechnology is based upon. One is genetic engineering and another is chemical engineering. What is a genetic engineering? It is nothing but alternation of chemistry of DNA and RNA to introduce the host organisms to change its phenotype. That is genetic engineering. Whereas this chemical engineering, what does it do is maintenance of the sterile ambience to enable the growth of the desired microbes or eukaryotic cells in large quantities for manufacturing of biotechnological products like vaccines. And then enzymes, beverages, drugs are also called as chemical engineering. Whereas genetic engineering, what is that?
Change the phenotype of the DNA.
Yeah, RNA. But whereas chemical, what is it? Yeah, sterile ambience like vaccines, in the preparation of vaccines, and then beverages and drugs. Okay. Now let us see what is a genetic engineering is all about. It is also known as recombinant DNA technology, and which means altering the gene in the living organisms to produce a new genotype. And there are various kinds of genetic modifications, like how inserting a foreign gene from one species to another. Okay. What does that mean? What is that? Inserting a foreign gene from one species to another. ஒரு ஜீன்லேருந்து ஒரு அந்த ஜீன் எடுத்து இன்னொரு ஸ்பீஷீஸ்க்கு இன்ஜெக்ட் பண்ணுறது, இன்சர்ட் பண்ணுறது, that is that. And altering an existing gene so that product can be exchanged. Okay, இருக்கிற ஜீனை கூட exchange, பண்ணிக்கலாம், altering. And changing gene expression so that is translated more often or not at all. Okay. Now let us see what is a genetically modified organisms. What is that? Kind of that. Yeah, that is our எந்த organismsலாம் நம்ம ஜீன் மாற்றி இது பண்ணுறோமோ they are all called as what? Genetically modified organisms. Is an animal, plant or microbes even. Okay, யாரெல்லாம் அதெல்லாம் we can change animals, plants, even microbes whose DNA has been altered using genetic engineering techniques. Okay, there are three basic steps in genetically modified organisms, like which include means introduction of the identified DNA. First one, எந்த DNA வை நம்ம identify பண்ணணும், first one, and then into அப்புறம் அந்த என்ன பண்ணோம், into a suitable host அந்த ஒரு DNA எடுத்து திருப்பி நம்ம replace பண்ணணும். That is the, what is that? Genetically modified organisms. Okay. Now let us see the construction of an artificial recombinant DNA molecule. Scientists Stanley Cohen and Herbert Boyer in 1972 isolated an antibiotic resistant gene and linked it to a plasmid, that is self-replicating circular extra chromosomal DNA of a Salmonella trypamurium. What is that? Salmonella trypamurium. என்னது அது? Yeah. என்ன பண்ணாருன்னா அவங்க ரெண்டு பேரும் என்ன பண்ணாங்க, ஐசோலேட்டட் ஆன்டிபயோட்டிக் ரெசிஸ்டன்ட் ஜீன். ஓகே, அண்ட் விச் லிங்க்ட் இன்டு அ பிளாஸ்மிட். அது என்னோட ஜீன், அது சால்முலா டைஃபையோட இது. அண்ட் நெக்ஸ்ட் என்ன பண்ணாங்க, கட்டிங் ஆஃப் த டிஎன்ஏ அட் த ஸ்பெசிஃபிக் லொகேஷன். ஓகே, ஸ்பெசிஃபிக் லொகேஷனில் அந்த டிஎன்ஏவை அங்கே கட் பண்ணிவிட்டு தென் த கட் பண்ண டிஎன்ஏ யார் கூட லிங்க் பண்ணாங்க, பிளாஸ்மிட் டிஎன்ஏ. அண்ட் தீஸ் பிளாஸ்மிட் டிஎன்ஏ ஆக்ட் ஆஸ் அ வெக்டர்ஸ் ஹியர். அண்ட் தென் லிங்கிங் ஆஃப் த ஆன்டிபயோட்டிக் ரெசிஸ்டன்ட் ஜீன் வித் த பிளாஸ்மிட் வெக்டர் டிஎன்ஏ ligase enzyme. And then RNA DNA is transferred to the E. coli and a number of copies formed by the replication, that is cloning. Okay. Okay. Now let us see the tools for the recombinant DNA technologies. There are various tools. Okay, like first one is enzymes, and then cloning vectors, and another is competent host organisms. Now let us see each one by one. Okay, the first one is enzymes. There are various enzymes involved in genetic engineering. The main enzyme is restriction enzyme. Where is? Yeah, restriction enzyme. Restriction enzymes they are known as molecular scissors. என்ன பண்ணுவாங்கன்னா, these are used for cutting the DNA at a specific locations. So, they are called as restriction enzymes. And these are of two types. One is exonuclease and another is endonuclease. Exonuclease is another. Yeah, remove nucleates from the ends. Ends la pandrathu exo. Endo na in the specific location. Okay, and the next enzyme is DNA ligases. DNA ligases are otherwise known as molecular glue. Okay, and what restriction enzyme otherwise known as? Okay, இவங்க வந்து molecular glue. இவங்க என்ன பண்ணுவாங்கன்னா these enzymes repair the broken DNA. Okay, ஏன்னா by joining two nucleates, அந்த பேர் என்ன சொல்லுது molecular glue. So என்ன பண்ணுவாங்க broken DNA they will be joining together. Another enzyme here is alkaline phosphatase. In the alkaline phosphatase, what they do is they remove the enzymes the phosphate group. Phosphate group la irukka enzymes they'll remove. That is known as alkaline phosphate. Another enzyme is DNA polymerases. Okay, DNA polymerases. What they do is these enzymes help in the vitro synthesis of complementary DNA strand on the DNA templates. Okay, they help in the in vitro synthesis, this DNA polymerase. Okay, and the next is cloning vectors. Okay, what is mean by a vector? Vector.
Vector carries.
Okay, a DNA molecule that can carry a foreign DNA, and what they do is that they replicate in the host, and that is called as vector. Okay, and there are three types of vectors. One is plasmid, bacteriophages and cosmids. What is plasmids? They are small and replicating usually in circular in shape. Okay, and this type of cloning occurs in many bacteria and yeast, and whereas bacteriophage, these are the viruses that will infect the bacteria. Okay, bacteriophages and all, and they are used as a vector because of very high number of copy of their genome within the bacterial cells. Okay, they have a high number of copy of what? Genome. Yeah, within the bacterial cells. Okay, now let us see the essential features of cloning vectors. There are two main things. One is origin of the replication and the second one is selectable marker. Okay, origin of replication means sequences that permit the propagation of itself in the host cell, and a cloning site to insert a foreign DNA. Cloning site la we have to insert the foreign DNA, and then the vectors contain a site that can be cut by a unique restriction enzyme. Okay, what is restriction enzymes? Molecular scissor cutting. Yeah, it's correct. Two types of restriction scissors. Restriction enzymes. Endo na? Inside varadu. Okay, avanga vandu in the specific region. Ana whereas that another is exona at the end of nucleotides of yes. Okay, and the selective markers enna pannum to select the host cell containing a vector with a foreign DNA, usually accomplished by a selectable markers for drug resistance. Okay, and the last one is competent host, that is for the transformation with recombinant DNA. Okay, DNA is a hydrophilic molecule. Hydrophilic na? Yes, so it cannot pass through the cell membrane since it is a hydrophilic in nature. So in order to force the bacteria to take up the plasmid, the bacteria cells have to be made competent na suitable, efficient avangala prepare paniknom. So there are some methods like micro injections and biolestics. Micro injection are what? They directly inject the DNA into the nucleus of an animal cell, but whereas biolestics, it is a gene gun, nothing but gene gun. What is that? They are bombarded with a high velocity microparticles of gold or tungsten coated with DNA. Okay, Okay. Okay, and the next, sorry. Okay, next one is recombinant DNA technology process. We'll see each step by step. Okay, first step na treat the foreign DNA and a plasmid with restriction enzyme and a DNA leakage. என்ன பண்ணுவாங்க அந்த சைட்டை வந்து treat a foreign DNA தனியா எடுத்துட்டு and plasmid with restriction enzyme ஓட work என்ன to cut the specific locations. So first வந்து என்ன பண்ணுது site where restriction enzymes cuts அது வந்து என்ன tDNA and this is plasmid and this is a what is that DNA. Okay, and the next step enna பண்ணுது DNA containing the gene of the interest இது வந்து it is inserted, it is உள்ள போயிட்டு அப்புறம் again what does it inserted DNA carrying the new gene நம்ம insert பண்ண T gene என்ன பண்ணிட்டு புதுசா ஒரு new gene உருவாயிடுச்சு அதுக்கப்புறம் that specific gene is treated with the plant and plant with a new trait whatever we want like desired traits can be done. Okay, this is the basic recombinant DNA technology process. Okay, now let us see the stages of recombinant DNA technology. The first process is isolation of the genetic material. What does we do here is most of the eukaryotic cells have DNA as a genetic materials as we all know, and then to form our DNA, the DNA should be pure, that is free from biomolecules or macromolecules. To form our DNA, what should be? DNA should be very pure. Okay, and this DNA located inside the nucleus enclosed with a plasma membrane and genetic genes intertwined with the histone protein. What we have to do is the cell have to break open to release the DNA and other macromolecules, and cells are lysed by lytic enzyme. What is lysed? Lysis nanadu? Cells are lysed. Separate. Lysis na separation, breakdown anything. And then lysosome, cell lysosome in the case of bacteria, and plant cell it is cellulose, and fungus it is chitinase. Okay, and the macromolecules treated with a specific enzymes to remove RNA ribonuclease and proteins as protease. அப்புறம் இதெல்லாம் முடிஞ்சிட்ட பிறகு the purified DNA precipitates with the addition of ethanol appears as a collection of the fine threads in the suspension. After all these process, last நம்ம DNA என்ன வச்சு இது பண்ணனும் ட்ரீட் பண்ணனும் ethanol. Okay, and the next process is cutting the DNA at the specific locations. By agarose gel electrophoresis, DNA undergoes a restriction enzyme digestion and cutting of the gene of interest and the addition of the என்ன பண்ணா நம்ம எந்த ஜீனுக்கு we are interested we should cut it and add what DNA ligase and the recombinant DNA is formed. Okay, and the next step is separation and isolation of DNA fragments. As a result of fragments of DNA separated by gel electrophoresis, that is previous process, depends on the negative nature of the DNA. Appuram what they do is external electrical field is applied to the medium to DNA move to anode, and then medium matrix used is agarose gel, that is a natural polymer extracted from the seaweed, and medium acts as a sieve and separate DNA according to their sizes, smaller fragments further, and then separated DNA enna pannum stained with ethidium bromide and exposed to UV radiations, bright orange color bands of the DNA in gel. Okay, and then after that ipo the stain bromide vechi pannita peragu separated DNA of the separate bands of DNA cut out from agarose gel and extracted from a gel piece, that is called as elution. And last is purified DNA fragments construction recombinant DNA by joining them with cloning vectors. Okay, and the next step is amplification of gene of interest using PCR. Okay, what is PCR? Yeah, and it is very best defined as it in vitro process of DNA replication. This technique was developed by Kari Mullis in 1985, who received a Nobel Prize for this. In 1993, he received a Nobel Prize and he discovered it in 1985. Okay, there are 4 basic requirements for this PCR technique. The first one is DNA template. What they do is total genomic DNA isolated from an organism that contains a target region to be amplified. Okay, and the next requirement is DNA primers. What are DNA primers? They are the short pieces of the single strand DNA. See, அங்கிருந்து கொஞ்சம் கொஞ்சம் இது இருக்குல short, they are the primers that flank the target. Another is stacked DNA polymerase. What they do, attaches the nucleotides on the growing strand of DNA. And the last one is nucleotides. They are polymerase adds complementary nucleotides to the template. Okay, these are the 3 main steps of PCR. Okay, the first step is denature the DNA. That is at 95 degrees Celsius. DNA is denatured, that is 2 strands are separated. And the second step is primers anil at the at 40 degrees Celsius to 65, the primers anil their complementary sequences on the single strand of the DNA. They bind with the complementary sequence, that is the second step. And the last step on the DNA polymerase extends the DNA chain. At 72 degree only, they extend the DNA chain by adding nucleotides to the third end of the primers. And the next process here is ligation of DNA fragment with vectors. DNA ligation is the act of joining together the DNA strands with covalent bonds with the aim of making new viable DNA or a plasmid. Okay, and there are currently 3 methods of joining the DNA fragment. The first one is, what they do is the DNA ligase that covalently joins the unhealed cohesive ends produced by the certain restriction enzymes, and the helpless they will join. Otherwise, that depends upon the ability of the DNA ligase from the phage T4 infected E. coli to catalyze the formation of phosphodiester bonds. Okay, and the next step is insertion of the recombinant DNA into the host cell. This is the last stage we came. What they do is, this occurs by several methods, but before which the recipient cells are made to competent na, like efficient, like ability. Okay, cells are made to receive the DNA. DNA receive ஆகிறதுக்கு அவங்க இதுவா இருக்கணும், efficient இருக்கணும். Okay, if a recombinant DNA bearing gene for the resistant to an antibiotic, that is example we'll keep ampicillin, is transferred into a E. coli cells, and the host become transferred into the ampicillin resistant cells only. So the ampicillin resistant gene in this case is called a selectable marker, and when transform cells are grown in the agar plates containing ampicillin, only transforms will grow and others will die. Okay, and the next step is obtaining or culturing the foreign gene product. Any protein encoding gene, what they do is expressed in a heterologous host called as what? Recombinant protein. Okay, these cells harboring cloned genes are grown in a small scale in a laboratory. These cloned genes are grown in a small scales in laboratory, and these cell cultures are used for extracting the desired protein using various separation techniques. Okay. The next one is bioreactor. Bioreactor na, these are the large volume vessels, approximately 100 to 1000 liters. The capacity is 100 to 1000 liters, and in which raw materials are biologically converted into a specific products. Biologically products converted into a specific products. Yes. Using what? Microbial, plant, animals or human cells only. Okay. Now let us see the components of bioreactors. The first one is an agitator system. okay, the agitation system, and then an oxygen delivery system, and then a foam control system. Oxygen delivery system iruka? Okay, first one agitation system, oxygen delivery system, and then a foam control system, appuram a temperature control, a pH control system. I think inga irukku nenikira ido. Okay, pH, temperature, aeration control system, and then a sampling port to withdraw the culture, a sampling port. Okay, these tanks are the choice of many, more than 70%, though it's not the best. Okay, and this have a following functions like what? Homogenization, suspension of the solids, and dispersion of the gas liquid mixtures, aeration of liquid, and heat exchange. This provided with a baffle and a rotating stirrer is attached either at the top or at the bottom of the bioreactors. Okay, a baffle of rotating stirrer is present either on the top or on the bottom. And then baffles, these are usually flat vertical plates whose width is about 1 to 10th of the vessel diameter, and normally 4 to 6 baffle plates are fitted to the individual vessel's wall to aid mixing and mass transfer the increasing turbulence. Okay, and preventing the vortex formation and eliminating what? Dead cells, dead spaces. Okay, the mostly used bioreactors are called as stirring type, and the stirring bioreactors are of two types. One is simple, another is packed. Simple and spragged, S-P-A-R-G-E-D. And the commonly used type is stirring type. Okay, and the last one is downstream processing. Downstream processing, na, after the completion of the biosynthetic process, the product has to be subjected to through a series of process before it is ready to marketing as a finished product. Finished product ku முடிக்கிறதுக்குள்ள ஒரு முன்னாடி ஒரு stage, that is the downstream processing. Okay, it includes two processes. One is separation, another is purification of process. It is seen in the diagram, that is cell lysis, separation, and then it is purified, and the pure supercoiled plasmid DNA. Okay, after this the product is then subjected to a quality control and quality control testing and kept in a suitable preservatives. After this downstreaming process.